Welcome to an overview of protein expression, where we'll connect strategic choices to practical outcomes. This presentation walks through how genes become products, which platforms make that efficient, and the process steps that turn laboratory expression into reliable production. The cover visual hints at workflows and data-driven decisions. We'll unpack slide by slide for a clear, applied perspective. This diagram reminds us of the central dogma. DNA is transcribed into mRNA and then translated into protein, and each step is a control point. Transcriptional regulation and promoter strength set how much message is made. RNA stability governs how long it persists, and translation rules like ribosome binding and codon usage influence final protein yield and engineering opportunities. E. coli is often the workhorse for expression, because it grows fast, gives high yields, and keeps costs low. Choosing the right strain, plasmid copy number, and induction strategy helps avoid inclusion bodies and maximizes soluble product. With optimized protocols, you can reach gram per liter titers within hours, making E. coli ideal for rapid, high-throughput production when simpler folding requirements apply. When targets need complex folding or human-like modifications, eukaryotic hosts come into play because they support disulfide bonding, glycosylation, and secretion. Yeast systems like Pichia offer microbial speed with eukaryotic folding, while HEK293 cells enable authentic post-translational modifications. Platform choice depends on target complexity, downstream application, and regulatory demands, so selection must align biology and end-use requirements. Optimizing expression requires matching the flux of protein synthesis to folding capacity. Promoters tune transcriptional flux, RBS sequences control translation initiation, and combinatorial synthetic biology tools let you test matched combinations. The goal is precise flux matching, so translation pressure doesn't overwhelm chaperones, minimizing aggregation and enabling rapid, stable production through rational screening rather than trial and error. Affinity tags and solubility partners are practical levers. Tags simplify capture during purification, solubility partners enhance folding and reduce aggregation, and engineered cleavage sites let you remove tags for therapeutic applications. There's a strategic trade-off between purification gain and preserving native structure. So small, minimally disruptive tags often strike the best balance for downstream functionality and structural integrity. Codon usage matters because ribosome speed and mRNA stability are tightly linked to translation outcomes. Harmonizing codon adaptation to host tRNA pools can prevent stalling and degradation while boosting soluble yield. Modern algorithmic gene design also optimizes GC content, mRNA secondary structure, and removes cryptic sites, producing sequences that translate smoothly and raise the chance of expressing properly folded protein. At the bioprocess level, control of metabolic burden through feed rate, cell density, and induction timing keeps cells healthy and productive. Soluble fraction is improved by temperature shifts and staggered induction strategies that give folding machinery time to catch up. Integrating online monitoring and automation enables reproducible, scalable bioprocesses, turning lab protocols into reliable production with minimal variability across scales. A simple, effective purification strategy uses two main steps: affinity capture to remove host contaminants, followed by size exclusion polishing to resolve aggregates and truncations. Process control, buffer pH, ionic strength, and gentle conditions helps maintain native conformation during capture and polish, yielding a final product with very high purity, suitable for research or therapeutic use while keeping structural integrity intact. Quality control ties everything together by verifying purity and consistency with orthogonal methods like SDS-PAGE, SEC, and mass spectrometry, then confirming biological activity through functional assays and quantifying contaminants such as endotoxin and host cell proteins. Rigorous data analysis and release criteria decide whether a batch is released or rejected, ensuring reproducible performance and confidence for downstream use. Different platforms support distinct product types: monoclonal antibodies, vaccines, enzymes, and high-titer production above 5 grams per liter can dramatically reduce costs and expand access. Platform adaptability is illustrated by case studies like insulin in prokaryotic systems and therapeutic antibodies in CHO cells, showing how matching platform strengths to product needs unlocks scale and global availability. Industrial enzymes, such as cellulases, proteases, and lipases, power large markets from biofuels to detergents and food processing. Directed evolution and high-throughput expression screens drive an innovation engine that yields pH-stable and temperature-resistant variants, delivering improved performance under extreme conditions and creating substantial economic and sustainability value across industrial supply chains. Cell-free protein synthesis decouples production from living cells, enabling rapid expression of complex or non-canonical proteins and direct integration with machinery and downstream processes. Continuous flow reactors with immobilized ribosomes scale this approach for industrial throughput, offering strategic advantages for on-demand biologics, personalized medicines, and vaccines by simplifying manufacturing constraints and shortening time to product. Bringing machine learning and cloud platforms into the workflow accelerates optimization by turning sequence data into predictive models for yield, solubility, and aggregation risk. Coupling ML with automated DNA assembly, robotic screening, and real-time analytics in cloud-integrated pipelines compresses optimization timelines from months to days, enabling rapid iteration and minimal manual intervention while maintaining data-driven decision-making throughout development.